Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I am Dr. Muhammad Ali Hashmi from University of Education Atak campus, and we are studying the subject of Introduction to Biochemistry. This is lecture number 18. It is about introduction and classification of lipids. The main concepts we are going to learn in this lecture are we are going to introduce the lipids, what are lipids, and then we are going to classify them into different classes and see their general properties. So first of all, what are lipids? We are studying biochemistry, so we are going to discuss the lipids biologically. Biological lipids are a chemical diverse group of compounds, the common and defining feature of which is their insolubility in water. So if you look at the biological lipids, the main property which distinguishes them from other chemicals is that they are not soluble in water. There are many substances in the human body or in the biological organisms which are soluble in water, but the lipids have the characteristic that they are not soluble in water, and they have a very diverse chemistry and diverse biological functions. There are so many types of lipids. There are so many different groups attached to them, so their chemistry is very diverse. And similarly, they have a lot of different biological functions which they perform in the living organisms. So we are going to discuss them one by one. Fats and oil are the principal stored forms of energy in many organisms. As you know that the fats and oils, they are actually the energy storage reserves which are present in many different organisms. So they have the stored energy which can be used for when the body needs it. Next, we see that lipids are very important components of the biological membranes. That means that they are components of the membranes which surround different like organelles, cells, etc. So what are membranes? Membranes actually they function to organize biological processes by compartmentalizing them. What does that mean? If you look at the cell, for example, it has many different organelles. So if there are no membranes around those organelles, what would happen? Everything will just mix up in the cytoplasm, and it will become like a solution, and there will be no organelles. So what differentiates an organelle from the other? It's its outside layer, which is its membrane. That membrane actually makes different compartments like nucleus, Golgi bodies, mitochondria, etc. And then these organelles can function on their own. They have different functions which they perform independently without mixing with the others. So membranes are very important to compartmentalize these organelles and organize them, organize different processes. So if you look at the cell, that is the basic unit of life. It is defined by its own envelope, which is the plasma membrane. So cell is defined due to this plasma membrane. If there is no membrane, then there will be no cell. Everything will be just burst out, and it will be no more defined as a cell or a unit of life. And if you look in eukaryotes, so in prokaryotes, the cell is defined by a plasma membrane, but in eukaryotes, not only the cell is defined by a membrane, but it has many other organelles like nucleus, mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies, and many other membrane-bound organelles. These all have membranes around them. So biological membranes, they are organized assemblies of lipids and proteins with small amounts of carbohydrates. These membranes, which we just talked about, the cell membrane and the membranes of the organelles, uh, they, their composition is the following. Like they have lipids and proteins in them. They are the major constituents of the biological membranes, and there are also carbohydrates in those membranes. So that is their chemistry. At the same time, we may say that these biological membranes, they are not impermeable barriers to the passage of materials. Like they do not let everything pass through them. They are not there just to let everything flow through the cell. So what they do, they actually regulate the composition of the intracellular medium by controlling the flow of nutrients, waste products, ions, etc. into and out of the cell. So what they do, they actually look for the materials and allow them selectively. मतलब वो हर चीज़ को सेल के अंदर या सेल से बाहर नहीं जाने देती। मेम्ब्रेन्स जो होती हैं वो कुछ चीज़ों को सेल के अंदर जाने की इजाज़त देती हैं, इस प्रोसेस को रेगुलेट करती हैं, और वेस्ट प्रोडक्ट्स को सेल से बाहर लेके जाने का भी एक प्रॉपर मैकेनिज्म है. सो दिस ऑल रेगुलेशन इज डन बाय द membranes. And how do membranes achieve this? How do they do this? They do this through membrane embedded pumps and gates. So there can be several pumps and gates in the membranes which are used to transport different substances against an electrochemical gradient or permit their passage with such a gradient. So substances can be allowed or rejected based on their chemistry, or based on their shape, or there might be different criteria on which these substances can be allowed inside to go inside the cell or go outside the cell. So this whole process is regulated through these membrane bound lipids. We are going to discuss this mechanism of the transport in the future lectures, how actually these membrane lipids allow or block certain things from entering the cells. So many fundamental biological processes occur on or in a membranous scaffolding. If we look at different biochemical processes occurring inside the cell, they actually occur either on a membrane or inside a membrane. That is very important. For example, if you look at photosynthesis, what is photosynthesis? Photosynthesis is the process in which light energy powers the chemical combination of water and carbon dioxide to form carbohydrates that occurs in plants. And where does it occur exactly in plants? It occurs in the inner membranes of the chloroplasts. So chloroplast is the organelle in plant cells where the process of photosynthesis happens. And due to photosynthesis, plants are able to use the water and the carbon dioxide in the presence of sunlight to form their food and the food for us. So this photosynthesis actually happens inside the inner membranes of chloroplast. Similarly, the processing of information such as sensory stimuli or intercellular communications is generally a membrane-based phenomenon. I mean, how does cells communicate with each other? That happens through these membranes. You know that neurons, they pass information or signals, so that also happens through their membranes, and membranes have a very important function in the body. Similarly, we are looking at the importance of the membranes. If you look at the nerve impulses, they are mediated by nerve cell membranes, just as I told you about the neuron cells, that they mediate their impulses through the membranes. And there are, of course, other substances like hormones and other nutrients, which are also detected by specific membrane-bound receptors. So membranes have a great importance in all these processes occurring in the body and inside the cells. So in this lecture and the next 2 or 3 lectures, we are going to examine the compositions, structure, chemistry, and the formation of biological membranes and their related substances, and the importance of lipids inside these membranes as we go forward. So we come to the lipids. What are lipids? Lipids is derived from the Greek word lipos, which means fat. They are the substances of biological origin that are soluble in organic solvents such as chloroform and methanol, but are only sparingly soluble, if at all, in water. What does that mean? That lipids are the biologically originated substances which were derived from the living organisms, and they have the characteristic property that they are soluble in organic solvents. For example, their lipids are soluble in chloroform, they are soluble in methanol, but they are not soluble in water. If what does this definition say? That they are only sparingly soluble if at all. अगर वो थोड़े से भी soluble हों तो वो बहुत sparingly soluble होंगे water के अंदर. Otherwise they are not soluble in water. So what is the advantage of this thing, that they are not soluble in water and soluble in organic solvents? That they can be easily separated from other biological materials by extraction into organic solvents, and then they may be further fractionated by such techniques as adsorption chromatography, thin layer chromatography, etc. कि हम इन्हें किसी ऑर्गेनिक सॉल्वेंट में डिसॉल्व करके इजीली सेपरेट कर सकते हैं बाकी सब्सटेंसेस के निस्बत, और तब हम फिर इसके अलावा जो डिफरेंट टेक्निक्स हैं लाइक एडसॉर्प्शन क्रोमेटोग्राफी एंड थिन लेयर क्रोमेटोग्राफी, कॉलम क्रोमेटोग्राफी, इनकी मदद से हम इनको completely सेपरेट कर सकते हैं ताकि हम इनको स्टडी कर सकें. Fats, oils, certain vitamins and hormones, and most non-protein membrane components, they are all lipids. So you see the importance and occurrence of lipids in the body, that all the fats, they are lipids, oils, they are lipids, certain vitamins are lipids, many hormones, they are lipids, and the components of the membranes, which are not proteins and carbohydrates, they are lipids. So lipids have a vast importance and different functions in the living organisms. Now we move on to the classification of lipids. We can classify lipids into following 5 different classes. Number 1 is the fatty acids and triacylglycerols, then glycerophospholipids, sphingolipids, and cholesterol. So in this lecture we are going to discuss all these 5 components one by one, and we'll see their main features about these classes. So the first one is fatty acids. What are fatty acids? As the name implies, fatty. What does that mean? There will be some fat portion. And acids, there will be carboxylic acids with long chain hydrocarbon side groups. So these long chain hydrocarbon side groups, they make the fatty part, and the carboxylic acid group is there to call it as an acid. So you can see on the right side here that these are different fatty acids. For example, stearic acid, you can see it has a 18 carbon long chain, and at the end it has a carboxylic acid group. So this long hydrocarbon chain is the fatty part, and the carboxylic acid is there at the end of the group. So the second one is oleic acid. It has a double bond in it. Similarly, linoleic acid, two double bonds, and alpha linoleic acid. It has three double bonds in it. So these are different fatty acid example. We'll see them in detail in the next slides. Fatty acids are rarely free in nature. I mean, they do not occur like this, like at their own in the nature, but they are bound with other chemicals. For example, they occur in the esterified forms as the major components of the various lipids. So this is a carboxylic acid, and it does not occur as it is, it occurs as the esterified form. You know that when carboxylic acids are they combine with alcohols, they form an ester, and these these fatty acids, they do not occur by themselves. Rather, this OH group makes an ester with an alcohol, and they occur as esterified forms. So here are some common fatty acids. For example, lauric acid, which has 12 carbons in it and 0 double bond. So this is a way of representing a fatty acid. We show its total double bond, its total carbon atoms, and then a colon and the number of double bonds. So these are all saturated fatty acids in which there are no double bonds. So for example, palmitic acid, it has 16 carbons in it and no double bond. But if you look at oleic acid, for example, it has 18 carbons in it and one double bond, and the position of double bond is n-9 means that it is written here, and it's the number of carbon atoms. And if you start counting it from the saturated side of the chain, and the double bond will be at the 9th carbon atom. We'll study about the nomenclature of fatty acids in the next lecture, and then you will see these things in detail, how we name them. So sometimes we also use a delta sign to represent the position of the double bonds in the fatty acids. So in the higher plants and animals, the predominant fatty acid residues are those of the carbon 16 and 18 species. That is the palmitic acid, oleic acid, linoleic, and stearic acid. So in most of the bigger plants and animals, the common fatty acid residues, they are of the following species, which are 16 carbon and 18 carbon atoms. And fatty acids with less than 14 or greater than 20 carbon atoms, they are uncommon, they are not common. So most of the fatty acid you will see or discuss in the living organisms, they will be between 14 and 20 carbon atoms. And one interesting thing, that most fatty acids, they have an even number of carbon atoms. Like you see 16, 18, 20. Why is this so? This is because they are usually biosynthesized by the concatenation of C2 units. So during their biosynthesis, at every step, they get 2 carbon units attached or added to the chain. So that is why the end product is the one having an even number of carbon atoms. So if you compare that, over half of the fatty acid residues of plant and animal lipids, they are unsaturated. What does that mean? That they contain double bonds and are often, they might be polyunsaturated. Polyunsaturated means that the fatty acid may contain two or more than two double bonds. So if they have only one double bond, we can simply say that they are unsaturated. But if they have two or more than two double bond, then we can say that the fatty acids are polyunsaturated. Bacterial fatty acids, they are rarely polyunsaturated but are commonly branched, hydroxylated, or contain cyclopropane rings. The fatty acids that occur in bacteria, they are mostly branched. That means they are not straight chain, they have branches on them, and they also have cyclopropane rings as substituents on them. And there are also unusual fatty acids which occur as the components of the oils and waxes. Um, that means the esters of fatty acid and long chain alcohols. We will study about the waxes in the next lecture. So these unusual kind of fatty acids are also produced by certain plants. Now we move on to the physical properties of fatty acids. The physical properties of fatty acids vary with their degree of unsaturation. So depending on the number of double bonds, their physical properties differ. The first double bond of an unsaturated fatty acid commonly occurs between its carbon number 9 and 10, counting from the carboxyl carbon atom. So this is a common thing, that first double bond usually occurs at the ninth carbon if you start counting from the carboxyl side. So that will be called a delta 9 or 9 double bond in terms of the nomenclature used for these fatty acids. We are going to discuss it in detail in the next lecture. So in polyunsaturated fatty acids, the double bonds tend to occur at every third carbon atom towards the methyl terminus of the molecule. So in polyunsaturated acids, it is usually observed that the double bond occur at every third carbon. For example, this is a chain. So if the first double bond is here, then the next one will occur at the third carbon. So if you start from here, 1, 2, and 3. So the next double bond is occurring at the third carbon. They are not conjugated. So double bonds in the polyunsaturated fatty acids are almost never conjugated. This is a conjugated double bond. Conjugated क्या होता है कि एक डबल बॉन्ड यहाँ हो, फिर एक सिंगल बॉन्ड और then double bond again occur करे. So for example, the bonds in the benzene ring, they are conjugated single and double bonds. But in polyunsaturated fatty acids, the double bonds are almost never conjugated. Triple bonds rarely occur in fatty acids or any other compound of biological origin. Saturated fatty acids are highly flexible molecules that can assume a wide range of conformations because there is relatively free rotation about each of their carbon-carbon bonds. So saturated fatty acids, they are flexible molecules. What is a flexible molecule? Which has single bonds in it. So organic compounds or molecules which have single bonds in them, they are flexible. They can rotate freely around those single bonds. So we call them flexible molecules. And the fully extended conformation of them is that of minimum energy, because this conformation has the least amount of steric interference between neighboring methylene groups. So they will occur in a position where the methylene groups are at a maximum distance apart, and there is no steric hindrance or interaction or interference between those groups. So we're going to discuss a little bit about the conformations. This is a very common example of N-butane. So if you look at the butane, this is the formula for it. And similarly, we can draw it in a different fashion, Saha's projection or the Newman projection. The Newman projection, the front carbon is presented by this dot and the back behind carbon is represented by this circle. So this is the first form of the butane, where these two methyl groups, they are anti to one another. They are exactly at 180 degrees to one another. But this is a single bond, and you see that these groups can freely rotate around this single bond. So if this conformation is taken and this group is rotated to this way, you see we end up in a conformation where this CH3 and hydrogen, they are eclipsed with each other. They're directly facing one above the other now. So this is a high energy conformation, and if we again rotate it, we end up at this conformation, which is a lower energy one. So we can get these different conformations of butane by rotating around its single bond between carbon number 2 and carbon number 3. So you should be familiar with this example as you have studied previous courses of organic chemistry. Um, you can study a bit more about conformations and familiarize yourself. Uh, the important point here is that the lipids, the saturated fatty acids, they can form different conformation, and they will be in a conformation which is of lowest energy. वो ऐसी conformation adopt करके रहना पसंद करेंगे जिसकी एनर्जी सबसे कम हो, ताकि वो stable रह सकें. The second class is the triacylglycerols. The fats and oils that occur in plants and animals consist largely of mixtures of triacylglycerol. Triacylglycerol is also called as triglyceride or neutral fats. They don't have any charges on them, they are neutral. These non-polar water-insoluble substances are fatty acid triesters of glycerol. So what is glycerol? Glycerol is this molecule. It has 3 hydroxyl groups attached to 3 carbon atoms to a propyl chain. So this is called glycerol. And if we react these 3 alcohol groups to 3 different carboxylic acid and make their esters, we make a triacylglycerol. So this is an acyl group, this is an acyl group, this is an acyl group. Triacyl, and the main component is glycerol. So triacylglycerols will be the mainly derived from this glycerol molecules. Triacylglycerols function as energy reservoirs in animals and are therefore their most abundant class of lipids, even though they are not components of biological membrane. Triacylglycerol biological membrane में नहीं पाया जाता, लेकिन फिर भी ये सबसे बड़ी एनर्जी reservoir है. ये as a stored energy organisms के अंदर मौजूद होता है. Triacylglycerols, they differ according to their identity and placement of their 3 fatty acid residues. So according to these 3 fatty acids we just saw on them, they differ in their identity. And the simplest triacylglycerol, they contain one type of fatty acid residue and are named accordingly. For example, if you look at this one, it has on all 3 carbons, it has the same component from stearic acid. So it has 3 components from stearic acid and the backbone of glycerol. So it will be called as tristearoyl glycerol or tristearine. So it has all 3 same acyl components. And the more common mixed triacylglycerols, they contain 2 or 3 different types of fatty acid residues. So the complex triacylglycerol will have these 3 acyl groups of different nature. They should not be necessarily the same. So we see the example on the next slide. Here is an example. You see that on the glycerol molecule there are 3 different kinds of fatty acid residues are attached. So at number 1 it is palmito-oleal, number 2 linoleoyl, this is from linoleic acid, and number 3 stearoyl, this is from stearic acid. So this will be called 1 palmito-oleal, 2 linoleoyl, 3 stearoyl glycerol. So the ones which are common are with different fatty acids, um, not the simple ones are so common in the organisms. Fats and oils. What is the difference between fats and oils? Fats are solid and oils are liquid at room temperature. That is the main difference. So, their other chemistry and the formula, they are much similar. These are complex mixtures of simple and mixed triacylglycerols whose fatty acid compositions vary with the organism that produced them. So ये जो fatty acids हैं इनकी composition depend करेगी कि किस organism में produce हुए हैं, उस हिसाब से ये vary करेगी. Plant oils are usually richer in unsaturated fatty acid residues than are animal fats, as the lower melting points of oils imply. So you see that we derive different oils from plants like sunflower oil, soybean oil, etc. Plant oils are richer in the unsaturated fatty acids. That is why they are liquid at room temperature compared to the animal fats, which are solid. Triacylglycerols, they are efficient reserves of energy, and this is because fats are less oxidized than the carbohydrates or proteins and hence yield significantly more energy on oxidation. If you look at the carbohydrates, they are more oxidized. They have so many OH groups on them, but fats are much less oxidized. So upon oxidation in mitochondria, they can they can yield much more energy than the carbohydrate molecules. And similarly, fats are nonpolar substances and they are stored in anhydrous form, mean they do not attach water with them. On the other hand, glycogen, which is a carbohydrate, it binds about twice the weight of water under physiological conditions. So ग्लाइकोजन या कार्बोहाइड्रेट्स को स्टोर करने के लिए साथ बहुत सारा वाटर स्टोर करना पड़ता है, जबकि फैट्स विदाउट वाटर स्टोर हो सकते हैं और मतलब लेस मॉलिक्यूल्स मोर एनर्जी प्रोड्यूस कर सकते हैं. In animals, adipocytes are the cells which contain fats. They are specialized cells which synthesize and store the triacylglycerols. The other types of cells only have a few small droplets of fat dispersed in their cytosol. Adipocytes, they are the main place where the fats are stored. You look at this figure, these are adipocytes which are filled with the triacylglycerols, and the entire cell is filled with those triglycerides. Adipose tissue that is made from the adipocyte cells is the most abundant in a subcutaneous layer. Subcutaneous layer is the innermost layer of the skin, which is also called hypodermis, and in the abdominal cavity. The fat content of normal humans, it is 21% for men and 26% for women. That is an average quantity that enables them to survive starvation for 2 to 3 months. That means that if we starve or don't have enough food for up to 3 months, we can use our fat reserves which are stored in our body for our energy needs. If you look at, in contrast, the glycogen supply, which is the short-term energy store that can provide for body's metabolic needs for less than a day. Like the glycogen, which is stored energy in our body as carbohydrates, that does not last long, that it just lasts for less than a day. That is why we have to eat more than once a day to meet our daily energy needs. So the subcutaneous fat layer that also provides thermal insulation, the innermost layer of the skin that has fats in it, it provides insulation to the body from the outside temperatures, and it is also an energy reserve. So this insulation is very important for warm-blooded aquatic animals like whales, seals, and penguins, which are routinely exposed to low temperature. So that subcutaneous fat layer protects them from the outside cold temperatures. The third one in the classification of lipids is the glycerophospholipids. Glycerophospholipids or phosphoglycerides. They are the major lipid components of biological membranes. So they are mostly found in the biological membranes. They consist of glycerol 3 phosphate. Here is the glycerol 3 phosphate structure. So this is the glycerol molecule, and on third carbon there is a phosphate group. That is why it is called glycerol 3 phosphate. When it is esterified at its carbon 1 and 2 positions, so this is carbon 1 and 2, these two hydroxyl groups, when they get esterified, and there is a phosphoryl group at the carbon number 3, then this class of compound called glycerophospholipids that comes into existence. So this phosphate group might have an X group attached to it at this position, and then carbon number 1 and 2 will have the fatty acids as esters on them. Glycerophospholipids are amphiphilic molecules. What does that mean? That means that they have two different sides, the hydrophobic and the hydrophilic side. It means they have water attracting and water repelling sides in them. They have the non-polar aliphatic tails and the polar phosphoryl heads. Look at this. These tails, they are non-polar aliphatic tails, and this phosphate group is the polar group. So they have both sides, polar and non-polar. That is why they are called amphiphilic molecules. The simplest glycerophospholipids in which the X is equal to H, they are called phosphatidic acids. They are present only in small amounts in biological membranes. In the glycerophospholipids that commonly occur in biological membranes, the head groups are derived from polar alcohols. We see the table on the next slide to discuss this. So this head group, this gets derived from polar alcohols. If it is simple H, it will be called a phosphatidic acid, and the H will be derived from water. If water acts as an alcohol, HOH, that looks like an alcohol, and then this will be the phosphatidic acid. If this X is derived from ethanolamine, this one, we have taken out the OH group, the alcohol group. So if that gets attached instead of X, that will be called as phosphatidyl ethanolamine. Similarly, if it is derived from choline, it will be phosphatidyl choline. So that is how the different glycerophospholipids will be having different X, which is derived from these alcohols. Glycerophospholipids are also named according to the identities of these fatty acid residues. For example, look at this example on the right side. I have colored these different portions to match according to the colors in the diagram, so you understand them very well. So if you look at this, on carbon number 1, it is the stearic acid residue. There is 1 stearoyl. On carbon 2 is the this portion is coming from the oleic acid, so 2 oleoyl. And on the third carbon is the phosphate group, but with phosphate the choline moiety is attached, so 3 phosphatidyl choline. You got how we name these according to the different residues attached on them. So some glycerophospholipids, they have common names. For example, phosphatidyl cholines are known as lecithins. What are these known as? Lecithins. The fourth one is sphingolipids. Sphingolipids are also the major membrane components. They are derivatives of the carbon 18 amino alcohols. Sphingosine, dihydrosphingosine. These are 18 carbon amino alcohols. Amino alcohol mean they have amine groups on them, and they also have alcohol group, and they have 18 carbons. So dihydrosphingosine is just the same part of the sphingosine with the saturated carbon atoms instead of one double bond in sphingosine. So sphingolipids, they are N-acyl fatty acid derivatives of sphingolipids are called ceramides. So if we make the nitrogen group acylated, that derivative of the sphingolipid will be called as ceramide. And ceramides only occur in small amount in plant and animal tissues, but they form the parent compounds of more abundant sphingolipids, which are described on the next slide. So ceramides are actually the compounds which further classify the sphingolipids. The first one is sphingomyelins. Sphingomyelins, they are the most common sphingolipids. They are the ceramides bearing either a phosphocholine or a phosphoethanolamine moiety. So they can also be classified as sphingospholipids. So if you look at this sphingolipid, it has a phosphocholine head group. It will be a sphingomyelin, the subclass of sphingolipids. Second one is the cerebrosides. Cerebrosides are the simplest sphingoglycolipids. They are alternatively called glycosphingolipids. They are ceramides with head groups that consist of a single sugar residue. If we look here, here the sphingosine and then a fatty acid residue, and then we have a galactose residue here, so it will be a galactocerebroside. So these will be of two types, galactocerebrosides and glucocerebrosides. Galactocerebrosides are the ones which mostly prevail in the neuronal cell membranes of the brain. They have beta D galactose head group, so that is why they are called galactocerebrosides. If we have a beta D glucose residue instead of galactose, then it will be called glucocerebrosides, and they occur in the membranes of the other tissues, not in the brains or the neuronal cells. Then we have cerebrosides. Cerebrosides, in contrast to the phospholipids, they lack the phosphate groups and hence are the most frequently non-ionic compounds. So as you've seen that the phosphate group is the ionic one, but the ones which lack the phosphate group, they will be mostly non-ionic. Hence the galactose residue of some galactocerebrosides are they are sulfated at their carbon 3 positions to form ionic compounds known as sulfatides. But some of them, they are non-ionic, but some of them may have a sulfate group. They will be called as sulfatides, so they will be ionic again. More complex sphingoglycolipids have unbranched oligosaccharide head groups of up to 4 sugar residues. So you see, if you keep increasing the sugar residues, the sphingoglycolipids, they keep getting complex. And the third one is gangliosides, the third type of sphingolipids. Gangliosides are the most complex group of sphingoglycolipids. They are actually ceramide oligosaccharides that include among their sugar group at least one sialic acid residue. In these ones, we don't have the simple sugars attached to them. We have oligosaccharides and we also have sialic acid residue in them. Gangliosides are primarily components of cell surface membranes and constitute a significant fraction of brain lipids. In brain lipids, about 6% are the gangliosides, and they also are present on the cell surface membrane. So we discussed the structures of 3 gangliosides, GM1, GM2 and GM3 here, which are out of the many hundreds known gangliosides. So here is the structure of the GM1 ganglioside, which is quite complex. As you can see, it has so if you look at this, if we break it from here, from this OH group, we put an H group with this O, that will be called a GM3 ganglioside. And if we break it from here, we remove this last D galactose moiety, it will be the GM2. This whole portion is the GM2 ganglioside, and if we do not remove anything, this whole structure is the GM1 ganglioside. So the sialic acid we described, here it is. Here the red colored moiety is the sialic acid residue. The gangliosides have considerable physiological and medical significance. Their complex carbohydrate head groups, which extend beyond the surface of cell membranes, act as specific receptors for certain pituitary glycoprotein hormones that regulate a number of important physiological functions. So their carbohydrate head groups, what are carbohydrates? These sugars, they extend beyond the surface of cell membranes, and they are receptors for certain hormones. So like gangliosides are also receptors of for bacterial protein toxin such as cholera toxin. Bacterial proteins also get attached to these gangliosides. The fifth and last class of the lipids is the cholesterol. Cholesterol is the member of steroids. So what are steroids? Steroids are the mostly of eukaryotic origin. They are derivatives of cyclopentano perhydrophenanthrin. So you see that this is actually a phenanthrene structure which is hydrated, and then it has a cyclopentane moiety attached to it. This is the basic backbone of the steroid nucleus. And the much defamed कोलेस्ट्रॉल जो कि बहुत बदनाम है, हम इसको मतलब बीमारियों से एसोसिएट करते हैं. It is the most abundant steroid in animals. It is further classified as a sterol. Sterols are those steroids which have an OH group at carbon number 3. And cholesterol has a specific side chain of 8 to 10 carbons at carbon number 17. The rest of the nucleus is the steroid nucleus. It is a major component of animal plasma membranes, where it is typically present at 30 to 40 mole percent. So in plasma membrane, cholesterol is 30 to 40 mole percent. It is a lot concentration. It occurs in lesser amounts in the membranes of their subcellular organelles. So it occurs mainly in the plasma membrane, but not in the, it does not occur frequently in the organelle membranes like mitochondria, Golgi bodies, endoplasmic reticulum, etc. So its polar hydroxyl group gives it a weak amphiphilic character. Due to this hydroxyl group, it can bind to water, but it has this whole hydrocarbon chain and other portion which is hydrophobic. So that gives it a amphiphilic character. That is why cholesterol is an important determinant of membrane properties. So this rigid nucleus also gives it strength. So it provides strength to the biological membranes. Cholesterol is also abundant in blood plasma lipoproteins, where 70% of it is esterified to long chain fatty acid to form cholesterol esters. So this OH group can be combined with the fatty acids to form esters. So this is cholesterol stearate. Stearic acid reacted with cholesterol to form this ester, which is known as cholesterol stearate. In plants, there is very little cholesterol. Instead, the most common sterol components of plant membranes, they are stigmasterol and beta-cetosterol, which differ from the cholesterol only in their aliphatic side chains. So these side chains are different for these two plant sterols, beta-cetosterol and stigmasterol. The remaining portion is similar to cholesterol, only the side chains differ from that. And in the yeast and fungi, they have a different version of the cholesterol that is known as ergosterol. It has an extra double bond here, and an extra methyl group at this position. So this is the summary of the lipids classification which we studied today. Lipids can be classified in the following main groups: fatty acid, triacylglycerol, glycerophospholipids, sphingolipids, and cholesterol. And then we also divided sphingolipids into three different classes: sphingomyelins, cerebrosides, and gangliosides. And cerebrosides are of two types: galactocerebrosides and glucocerebrosides. So this was all about the classification of lipids. And you are advised to visit the following links. They have very useful material. They have 3D structures of these lipids, which will help you understanding their conformations. And if you have any questions, you are free to ask in the Google Classroom. And these are the books which were consulted to prepare this lecture. I'll meet you in the next lecture. Until then, Allah Hafiz.